2 degree in Chandigarh.
Okay. Good morning, one and all present here. Myself, Dr. Pranita Gulane, on the occasion of 124th birth anniversary of Dr. Panjabra alias Bhausaheb Deshmukh, Shri Shivaji Education Society, Amravati Science College, Congress Nagar, Nagpur, in association with Microbiology Society India, organizing a lecture series on the theme Indians Speaking from Abroad. Today's lecture is lecture 19, which is on the topic magnetic nanoparticles in feces of humans with Parkinson's disease. So on behalf of the organizing committee, Honorable Principal Professor Mahendra Dhore Sir, Science College, Congress Nagar, Nagpur, and Honorable President, Microbiology Society India, Dr. A.M. Deshmukh Sir, I welcome our eminent guest speaker, Ms. Bidhi Dwivedi Madam, Research Assistant, University of Helsinki, Finland, as well as all the dignitaries and all the participants of today's guest lecture. I take this opportunity to introduce today's guest speaker, Ms. Bidhi Dwivedi ma'am. She is currently in Helsinki, Finland, as an research assistant. She has worked on the Parkinson's project in the Perseris University of Helsinki Department of Microbiology. In her bachelor's thesis, she has worked on in silico analysis of lifeguard proteins in Homo sapiens in 2020. Before it, she has worked on detection, quantification, and isolation of D-sulfovibrio species in the feces of Parkinson's disease patients and healthy controls in her post-graduation thesis. She has also done internship at Man Pharmaceuticals Limited at Gujarat, India. She has worked as a molecular biology intern at Indian Bioscience Institute of Technology and worked on the advanced molecular biology techniques at Delhi, India. She has her expertise in genome sequencing as a bioinformatics tools, as well as in biostatistics, restriction analysis, electrophoresis, mammalian cell cultures, qPCR, polymerase chain reactions, working with microbes in anaerobic environment, etc. She has also volunteered and spent time working with various NGOs, starting from her school days. At SRM, where she has completed her bachelor's degree, she has worked with an NGO called Blooming Beacon, which is recognized by the government of state of Tamil Nadu. As well as she is an successfully hosted various events and activities like blood donation camps. She has also organized checkup camps, food donation camp, tree plantations, and celebrations of festivities for the economically backward section. So thank you very much, Ms. Bidhi Dwivedi ma'am, for accepting our invitation. On behalf of the organizing committee, I welcome you in today's guest lecture series, which is based on the theme Indians Speaking from Abroad. So please elaborate your topic, ma'am, and please share your journey from India to abroad to our participants. Please, ma'am.
Yeah, sure.
Can I start? 1 minute.
Okay.
1 minute. First of all, I will make you co-host. You are co-host. Okay. You can share your PPT if there is a PPT. If you have.
Yeah, I have PPT. Like, can I start now?
Share.
Can I start now?
Yeah.
Yes.
Okay.
And in the meantime, I welcome all the participants today. The
My
reason
father
is
here in the participants, and I'm feeling a little bit nervous now.
Correct. And your lecture is being recorded.
Okay.
And it will be available to 20,000 students of Microbiology Society.
Wow.
Yes. And really your journey will be inspiring to our students. Ultimately, you are contributing for the development of India.
Yes, thank you very much.
Many stalwarts today are here, like Swarnajit Singh, like a Patankar sir, like a Prakash Halami, and other teachers are also there. Rakesh, who is working on nanoparticle, So like that, many stalwarts are there attending. Thank you all. Over to Vidhi.
Okay, so I'm Vidhi Divedi, and before we move further, I would like to thank you all for having me here. And it is such an honor for me to present myself before you all on the special occasion of 124th birth anniversary of Dr. Punjab Rao Deshmukh, which is happening at Sri Shivaji Science College in association with the Microbiologist Society. So today I'll be talking about the magnetic nanoparticles in the feces of humans with Parkinson's disease, which is basically the project that I'm working right now. So what we have done is like we have isolated the magnetic nanoparticles and a special type of bacteria from the feces of the people who are suffering with Parkinson's disease and 10 people who did not have any disease, the healthy controls. And I'll elaborate it in detail in my further slides. But before I go further, even though Dr. Pranita explained a little bit about myself, I would like to focus something about my background. So I was born in a very stubborn village of India, which is in Uttar Pradesh, if you guys know Gorakhpur. I was born on 13th August 2001, and obviously, I'm still an Indian by nationality, and I'm currently working as a research assistant in University of Helsinki. So I did my schooling in a small town near my village, but then I moved to Chennai for my bachelor's in biotechnology, and I did my graduation at SRM Institute of Science and Technology. I got my degree, bachelor's degree in the year 2020, and I got enrolled into my master's program at the same in the same year at University of Padua in Italy. And my master's program was in biotechnology for food science, and I was one of those students who were awarded with the scholarships provided by the Italian government. And this scholarship did not only cover my university fee, it also covered my personal expenses, also my accommodation. So I did not have to worry about my financial situation while I was doing my master's. And then I have done when I was in Italy, for some regions I did not find myself growing. I found that somewhere my growth is stuck, and even though University of Padua is like one of the best universities in the world, I was somehow not satisfied, and I went to the office and I said that the things that I'm learning here are not, like, I did not expect this. Obviously, when I'm leaving my country, I'm looking for something great. And this they were like, okay, they provided us with the special classes and stuff. And in the year 2021, the students from the University of Padua who were very good at their academics were provided with the Erasmus opportunity. Erasmus is basically a semester exchange program. So I got the opportunity where I could do my semester abroad, like not in Italy. I can do as an Italian student in some other country. And Finland is like the happiest country, obviously, but University of Helsinki is also one of the best universities in the world. And I got the opportunity to do my master's thesis here, and yet again, I was provided with the scholarship that covered my University of Helsinki fee, my accommodation, and my personal expenses. And apart from that, I have done hands-on training in all the advanced molecular biology techniques at Indian Biosciences in New Delhi. I also worked at Man Pharmaceuticals, where I was able to gain knowledge and experience on like the product development, how from the initial stage, how the product is made and how it is released into the market. And then I have certification in drug discovery. So basically, I was able to learn the drug discovery pipeline, for which I did this course at University of California in San Diego. And I have participated in international conference on nanotechnology at SRM Institute of Technology. For some regions, nanotechnology and nanoparticles have been a very crucial part of my life. And then when I was in third year of my bachelor's, I got the opportunity to do a poster presentation on nanobots. Nanobots are basically nano robots. So the main like what are the advantages of using nanobots was like we can use personalized medication, like we can use on site, we can do target I don't have proper word, like I'm just like a little bit nervous. So it's like basically if you have something, we can go on the site of the infection or the site of the disease and treat it on the site, and we do not have to involve all the body metabolism to treat one disease. And apart from that, I have been an active member of an NGO, and I have worked as a social activist. And the main aim of this NGO was to create awareness among the women of India about the menstrual hygiene, and because for some reasons we still have a taboo related to the menstrual hygiene. So we have created a lot of awareness. We were able to collect money, and we were able to uh, we did like people donated money, and we bought sanitary napkins, and we distributed in the people who are not aware of the menstrual hygiene, and they are not capable enough to buy sanitary napkins. So we went like from houses to houses, and we distributed the sanitary napkins, and we provided them with the knowledge that it's very important for them to take care of themselves. And when I was in my bachelor's, I worked as a coordinator, and I have coordinated in many cultural and sports fest. Uh, and that's it. That was all about my background. And now I'll talk about my current research project that I'm doing in Helsinki. So basically, as I told you, I'll be talking about the magnetic nanoparticles in Parkinson's disease. So from my study and from the magnetic analysis that has been done on human tissues have revealed that there are nano-sized ferrimagnetic magnetite particles present in brain, liver, spleen, heart, etc. And they are basically found in two different types. The first ones are the euhedral crystals, which are possibly of the biogenic origin, and the other one is the rounded crystals, which are of convention derived origin. So when I'm talking about convention derived origin, I'm talking about when we burn something, we are producing magnetite, and the biogenic origin is like the magnetite that is present in the sediments of the ocean and rivers and stuff. And it has been found that the all the magnetic properties of these magnetite particles is totally dependent on the magnetic relaxation time. The magnetic relaxation time of these magnetite particles is determined by the particle size, the morphology, the microscopic coercive force of the material, the magnetization of the sample, and the thermal energy. So basically, these magnetic nanoparticles are of two different types. The first ones are the one that are less than 30 nanometer in size, and they are called as super paramagnetic nanoparticles. And then there are the other ones which differ from 30 nanometer to 80 nanometer in size, and they are called as single domains. These were some quite general information about the magnetic particles, but we are going to know and discuss about what is the effect of these magnetic particles on the human body, how it is causing the disease. So it has been found that these magnetic nanoparticles have the capability that they can translate possible effects to the tissues, because the static magnetic fields can reduce the mitochondrial membrane potential. They have the capability to generate oxidative stress, and they have the capability to induce the process of apoptosis. So when we are talking about the reduction in the mitochondrial membrane potential. We are talking about the early, like when there is a reduction in the mitochondrial membrane potential, it is an early sign of apoptosis. And there are several studies and there are several papers which have found that the magnetic iron oxide, which is the magnetite, and the magmamite, which is the oxidized form, it has a very possible and important role. It might play a very important role in the pathogenesis of neurodegenerative disease, and Parkinson's is one of the neurodegenerative disorder. So I'm the thing, like iron and the magnetic nanoparticles, when they are present with the ferric ion, that is Fe3 plus ion, they have the capability to synthesize alpha synuclein, which aggregates into the central and the peripheral nervous system. And the formation of these aggregates or these toxic structures in the nervous system is a hallmark of Parkinson's disease. Like if you can determine or you can identify a toxic structure in the peripheral and central nervous system which is made from alpha synuclein, that means we can confirm that this person has Parkinson's disease. So before we go further, I will tell you a little bit about Parkinson's disease. It is basically a movement disorder. It is most prevalent movement disorder, and it occurs mainly in the elderly people. Like they do not have control over their body movement. They're shaking all the time. So it is a neurodegenerative disease where the alpha synuclein, a neuronal protein, it forms the aggregate, and it aggregates and it leads to the formation of toxic structures in the central and the peripheral nerves. And there are many studies that have been done on Parkinson's disease, and yet the real cause of Parkinson's disease is unknown and it remains unclear. But the intestinal dysfunction and the changes in the microbial gut biota is it's like my it has been linked with the pathogenesis of pathogenesis and disease progression. But we still, I mean, there are a lot of studies, there are hundreds of papers that are talking about the microbial etiologies, like how this there is a change in this microbial microbiota, there is change in this, there is change in this, but it doesn't give you a surety that maybe because of this microbe we have Parkinson's. So it's like we have a lot of information, but still we cannot ensure something with 100% confidence that this thing is responsible for the disease. So what I'm doing right now is we have found a bacteria, gram-negative sulfate producing bacteria from the genus D. sulfovibrio, which has a very potential role in the progression of the disease. Like right now, my entire work is focused on D-sulfovibrio, and we found out that these gram-negative sulfate-reducing bacteria have, like, they are capable of causing the disease. And so basically what we are doing right now, we took 10 healthy controls, the people with no Parkinson's disease, and 10 people who have the disease. And we have to assess that in order to determine that there is a relation between the D-sulfovibrio species which is present in the microbiota, in the gut microbiota. So what I have done, I have taken the poop sample, the feces of the patients and the feces of the healthy people, and I have isolated the DNA, and then I did a conventional and real-time PCR to find out the relation between the amount of DSV that is present, which is amount of D-sulfovibria which is present in the sample, and what is the severity, like is there a chance that people who have more bacteria have like severe Parkinson's symptoms. So we did our experiment in the university with different type of PCR, and we found out that uh, there was a difference in the like the severity of the disease is directly linked with the amount of bacteria that is present in the poop sample. And we have used different type of primers like HID A. We have used these. I can show you. So basically, we have found the samples with like these are healthy samples. We have used Roman numbers to sorry. We have used Roman numbers to in like to talk about the healthy people, and then the numerical numbers like 1 2 3 for normal, the mathematical numbers for the patients. And here you can see that we have found different type of desulfovibrio in different type of in different samples. Like our patient number 1 was identified with DSB fairfieldensis, which is a strain of the bacteria, and then the Piger, and then we have desulfuricants. I've okay, we have desulfuricants as well. So we have used six different primers, which is MB, Wolf, Fair, Pizer, PA, FV, Haid, to identify the different strains of bacteria. And we performed the experiment on only the samples, which were about like approximately 40 to 45 nanogram per microliter. So I have told you already that when the amount of bacteria was directly linked with the severity of the disease, and we found this severity, like the main reason why this like we have more symptoms of the Parkinson's in the people with more amount, the large amount of bacteria, is due to the fact that this DSV, which is the gram-negative sulfate reducing bacteria, has the capability to produce hydrogen sulfide and lipopolysaccharides, and several strains of this bacteria can synthesize magnetite, and they are eventually leading to the leading and inducing the oligomerization and aggregation of alpha-synuclein, which I told you before, which is a hallmark of Parkinson's disease. So the formation of alpha-synuclein protein is due to the oligomerization, which is like this bacteria induced the oligomerization by forming hydrogen sulfide and lipopolysaccharide, and by synthesizing magnetite. So the entire plan was to detect the presence of DSV in the feces of the patients and the feces of the people with no disease. And we use the protocol which were described in Muras et al. In 2021. So he has done his experiment on the magnetite particles that are present in the cervical skin. So he has basically done his experiment on determining the role of magnetic nanoparticles in Parkinson's disease, but our research is mainly on feces, and his research was on cervical skin. And then we quantified the amount of DSV. We used quantitative real-time PCR. And the first and second part of the experiment is done, but for the third part, we have given the samples to the to the geophysics laboratory, solid earth geophysics laboratory, and we are still waiting for the results. Meanwhile, I have done the statistical analysis, and the conclusion of the project is the conclusion of this entire project is that the more is the amount of DSV, the higher is the severity of the disease. And I still have a lot of work going on, and I should provide you with the results of the I should provide you with how much amount of magnetite we collected from samples, like the people who were suffering and people who were healthy. But unfortunately, everyone is on Christmas holiday, and I still don't know when I will be getting the results. But I hope I'll be getting it soon. And I'm publishing a paper next year. So if we are in contact with each other, you can read my paper and maybe you can find something useful from it. So this was all about my research. And if you will talk about my next 5-year plan, so I'm still a research assistant, and I am looking forward to pursue my PhD by next year, and I want to see myself as an expert in microbiology in coming 5 years. Thank you very much for having me again and listening to me.
Yes, ma'am. Thank you very much for your detailed information regarding your topic on contamination in means contamination of nanoparticles in feces of the people who have Parkinson's disease.
Yeah.
Yes,
If you
ma'am.
have any questions.
Yes, yes. If the participants have any queries, then they can put in the chat box. Yes, there is one question from Dr. Prakash Halami, sir. What type of microbiota are predominant in Parkinson's disease? How magnetic nanoparticles are associated with prevalence of Parkinson's? First question is, ma'am, what type of microbiota are predominant in Parkinson's disease?
Well, if I cannot explain all, but there are different like if I have to explain, like I told you, there are different type of bacteria that are producing different type of strains, like different organic, like the metabolites inside the body, which is like they're producing the toxins. The generation of toxins, and how they have the capability to reduce the sulfate inside the body. And I mean DSV and other strains. Like if I have to give you some names, I will tell you the Vericum microbiota, Bifidobacteriaceae, Crystal Cranliaceae, and these are scientific terms. So
Okay.
these are, if you want, I can write it in the chat.
Yes, yes, ma'am.
So that they can have a reference.
Okay. There is one more question from Dr. Prakash Alami, sir. How magnetic nanoparticles are associated with prevalence of Parkinson's?
Yes, I will explain that. Basically, they are affecting the mitochondrial activities, and these magnetic fields have the capability to induce cell death in inside our body. And for some regions, they have ability to synthesize and to form alpha synuclein, which is a neural protein. So when this protein is present in the brain, and it is aggregated in such a way that it leads to the formation of the structures that somehow interfere with the normal function of the neurons and the nerve cells, which eventually lead to the disease.
Okay. One question is on how was prevalence of DSV among Parkinson's patients? Does DSV is associated with magnetic nanoparticle synthesis?
Yes, they are. As I told you before, they have the capability to produce lipopolysaccharides. The DSVs are gram. So the entire idea about explaining and doing this project was to understand how they're affecting the body, like how the DSVs are capable of causing a disease to our body. So the idea here is we found out in our study that these DSV have the capability to produce lipopolysaccharides, and these lipopolysaccharides are more in amount, like they are in more quantity in the people who are suffering from Parkinson's disease. And these metabolites that are produced by the desulfovibrios have the capability to form the magnetites, to synthesize magnetites, and these magnetites eventually lead to the disease.
Okay.
So it's the DSV that produce the lipopolysaccharide, and then these lipopolysaccharides lead to formation of synthesis of alpha-synuclein, or it leads to formation of it leads to the synthesis of magnetite particles, which eventually interfered with the normal neuronal function, and then it eventually caused the disease.
One question is from Rakesh from Jambeshwar University of Science and Technology. He is asking that how to approach to your university for PhD admission.
Well, there are two different ways. There are open applications on the university website where you can directly apply and like go through the institutional process, and there is other way where you contact the professors on your own. Like for me, if I have to contact someone from India at University of Helsinki, I'll write them that, hello sir, my name is Vidhi Dwivedi, and I will explain them a little bit about my background. I will add my resume and a motivation letter, and obviously I'm going to read about that professor. Like I'm going to get a little bit knowledge about his background. So when you are writing an email, you have to write that you have studied, you have like this professor is working on this project, and you are interested in the project, as you have, like you have to provide them a link between your work and their work. And so if you are writing, you can write, hello, my name is Rakesh, and I want to do PhD. I'm looking for a PhD program. I'm from India, and I have done my bachelor's, master's in this thing, and I read about your research background, which closely aligns with my research background. And if you have any PhD position at the moment, I know a lot of people who get into PhD programs, and that are well-funded PhD programs this way. And obviously, like there is other way, the institutional way, where you go and you apply on the website and you wait for the like all the processes to be done and stepwise. But the direct way is to email the professors, and that's a very good, like like that's a very good approach, because maybe even if the professor doesn't have a PhD position at the moment or don't have the funding, maybe you can talk to them and you can ask if they can provide you with an internship, or they can allow you as like for a short term tenure in their place, and that way you can find your way to PhD.
Okay, okay. So
Hello,
thank you very
hello,
much, ma'am.
hello.
Yes. Yes.
One thing. Your lecture and your topic is very nice regarding
Thank
the
you very much.
Parkinson. One thing is there, as Dr. Halami also asked regarding the desulfovibrio DSV. Have you made any study that any dietary thing or any nutrient which is going through the nutrition of the person, it is supporting the DSV in the gut? Is there any association have you got in that direction?
Well, I would love to do something with the nutrition, with the association of, I mean, I would definitely do
Diet
some,
and the
yeah, I would
yeah.
definitely do something related to the nutrition or the dietary supplements that are enhancing the growth of the bacteria inside the body. But until now, we do not, we recently, this project is very new, so we are
Yeah.
still working on it, and there is a lot that has to be done,
Yeah.
and there is a lot that I should learn as well. So
But
it's like a very new project. But
that particular angle might be there which support the growth of DSV or the colonization of the DSV in your
yes, yes, but we recently found it out, so maybe in future you can find the papers that will provide you information on this, and it is possible that in future I might do something on this.
yeah.
And thank you, it was a very nice question, but we do not have any
Okay.
information about this right now.
Thank you.
Yeah.
Vidhi,
Yes.
You put your email ID in the chat box. Your email ID.
Okay.
It is a bidhi.dwivedi
Yes.
Isn't it?
That's my student email ID. So I would ask you all, if you have any queries, please contact me on my personal email ID, because
Yes.
I don't know where I will be, in which university by next year, so it
Okay.
might expire.
Okay.
Okay.
So
Thank
you
you
give
very much,
your
ma'am.
personal email ID. Give your
Yeah.
personal email.
Okay. So
Yes.
shall we conclude here, Dr. Deshmukh, sir?
Yes, you can conclude if there is no question
Yes, sir.
from
Yes.
remaining participant.
I hope
Anyone?
my presentation was clear and nice enough, and I answered all your queries well.
Yes, ma'am.
Yes, and that was a excellent and high level interactions.
Oh,
So
thank you very much.
yes, and it is expected. The reason is that, as I have told, Microbiology Society is arranging lectures every Sunday.
Oh, okay.
Yeah, and that is continued from 4-5 months back. And
Okay.
again, I will request other participants. You can join at 2 p.m. There is a one more lecture at 2 p.m. which is
Okay.
on how to obtain travel grant for international conference.
Okay.
Yeah,
Oh.
and Bidhi, you can give your WhatsApp number so that I will include you in one of our group.
Okay, sure. Sure.
So you will
Yes.
know all updates of Microbiology Society regular.
Okay.
Okay.
Yes. Thank you
Yes.
very much. I really feel honored that I have this opportunity to present little bit
Yes.
about myself in front of you all. Like,
And
it's such an honor.
you give your address, home address of India,
Yes.
so that Pranita ma'am will send you the memento of Microbiology Society on
Okay.
Yes,
your.
ma'am.
Okay.
Yes, ma'am.
Okay. Okay.
Pranita,
Thank you
you
very
can.
much, ma'am. Yes, sir. Yes. So please give your Indian postal address so that we could be able to send you the memento on it.
Is
And
it
thank you very
okay
much,
if
ma'am, for your great lecture and the information which you have given. This is a very good interaction on the said topic on magnetic nanoparticles in feces of humans with Parkinson's disease. So thank you very much, ma'am, on behalf of the organizing committee, Science College, Congress Nagar, Nagpur, and Microbiologist Society, India. We are very much thankful to you for accepting our invitation in the
same?
lecture series based on the theme Indian speaking from abroad. So thank you very much, ma'am, once again.
There are people asking for my contact number.
We will share it on the.
Yes. Yes. Okay. You can also contact me on LinkedIn. That's a more
Yeah.
professional way of
And
contacting the
yes,
students.
and with
Yeah.
your permission only, we'll
Yeah.
share
Yeah.
the.
Yeah. Yeah.
Yeah,
Yeah.
with your permission. Without your permission,
We will
no
not share.
contact. Yeah.
No,
Yes.
no, you can share, but it's just that, I mean, on LinkedIn, they can see what I'm doing or where I am at my
Correct.
life at the moment. So
Okay.
that's a more professional way of interacting with each other.
Yes.
Yes. Fine. Okay, ma'am.
Thank you
Thank
very much. And I'm mobilized again.
Yes.
Thank you.
Yes.
Thank you very
Merry
much.
Christmas, ma'am.
Yes,
Okay.
Merry Christmas to you too. I'll share my LinkedIn. One moment. I will share my LinkedIn in the.
Okay. Okay, ma'am.
Yeah, it is very simple to get your number at LinkedIn.
Mm-hmm.
I can also search, no problem. Okay.
Yes,
Okay.
yes.
Okay.
So link Bidhi
Yes,
Dwivedi.
yes, it's
You can.
Vidhi Divedi. For some reasons, I can't find the link, but I'll just write my name so that it will be easier for you
Yeah,
to find
yeah,
me.
yeah. You write down again your name in the chat box,
Yes,
so people will search you on LinkedIn. And
and it's University of Helsinki, so it will be easier for you to
yeah,
find me.
okay.
Okay,
Bye.
everyone, have a very nice Sunday and a very great day ahead. And for
Okay.
all the students here, I wish you all the best for your exams and your coming future ahead. and I will take a leave now. Bye bye.